champions, I welcome you all to score 360 out of 360 in NEET Bio within 250-day series, and today we are going to finish this chapter off. Yes, so today we are going to talk about seeds and the types of fruit, apomixis and polyembryony, and also artificial hybridization. So as I said, I won't be stretching this chapter, I am keeping the promise. And one more thing, we might have a rapid revision tomorrow, okay, for everything that we have studied. Going ahead, let's see what did we cover in the previous session. Yes, so we spoke about embryo and endosperm formation and also pollen-pistil interaction and double fertilization. If you want the notes, you can join the WhatsApp channel. It includes just us, the NEET people. Going ahead, number of questions, there were 2 into 4, 8 marks. Can we lose 8 marks? No, we cannot lose 8 marks. So this is why the chapter is important. Now, do you remember that I gave you a homework as well? Well, let's see who all got it correct. In a cereal grain, the single cotyledon of embryo is represented by. Now, single cotyledon, when the term single cotyledon comes, that means I can say that they are monocotyledons, or basically that this particular plant is what? A monocot. Now, the single cotyledon which is there in monocots, what do you call this as? You call it as scutellum, and hence C is the correct option. Okay, going ahead, let's start off with the seeds. I know a bit of it we covered in the previous session, but why not? We can cover it now also. Now, first thing. So basically, if I say that this is the stigma, just a second. Okay, just for you to revise. And this whole thing is the ovary, and inside the ovary, what do you have, children? You have the ovule. Now, agree? Ovary which is there, it will now form the fruit. Agree? The ovule which is there, it will now form the seed, and inside the seed, what do we have? Embryo. Okay, so inside the ovule you had two things initially. One is the embryo, or basically inside the seed you have the embryo, plus what do we have? Initially we had endosperm. So basically, when one male gamete fused with the female gamete, you got zygote, and zygote formed the embryo. Now the second male gamete, I am writing second male gamete, when it fused with the two polar nuclei which were in middle, it formed endosperm. Now endosperm is the one that is basically going to provide nourishment to the zygote to form the embryo. As of now I have written already embryo, okay, but initially it was zygote, it formed an embryo with the help of endosperm. Now there are certain seeds, okay, where the endosperm is totally utilized by the growing embryo. Endosperm is totally utilized by the growing embryo. It says that it'll give me everything I need. And when it is totally utilized, so I am saying it is totally utilized, at this point of time I say that there is no endosperm. Agree? No endosperm left. Agree? Now no endosperm as in, but it's a seed, it's still an embryo. It obviously needs some stored nutrients, or else it won't survive. We never know how and when the time will come when it will be finally able to germinate into what, a new plant. Okay, so it definitely needs some stored form of nutrient. So what does this these type of seeds do? The ones where the endosperm is not there, it is totally utilized. It is utilized for the formation of something which you call as cotyledon. Okay, so these type of seeds, they take all the endosperm for the formation of embryo. Now what is the embryo? Embryo will also have two cotyledons. So these two cotyledons are formed with the help of the endosperm. There is too much endosperm such that they form two cotyledons. Now this type of plants, I call them dicots. Or dicotyledons. Okay, going ahead, next thing I should be making it a bit nearer. So dicots. Now in the next case, not fully utilized. There will be a case where the endosperm is not fully utilized. Okay, the endosperm is still there, and it happens that little amount of endosperm which is being used, it is to form one cotyledon, and the plants that has one cotyledon, what do you call them? Monocots. This is the question that we discussed now. This is the question that we discussed now, and this single cotyledon, what do you call it? You call it scutellum. Apart from the scutellum, they will also have some endosperm left. As I said, it is not fully utilized. Some is utilized for making one cotyledon, some is utilized for just endosperm to be there. So endosperm is still being there. Now, since here there is no endosperm, I say it is non-albuminous. I say the dicots, basically they are non-albuminous. They have non-albuminous seeds, that is they do not have, you can say, non-endospermic. They do not have the endosperm left, they have used for making cotyledon. Now on the other hand, the ones that have cotyledon, only one cotyledon and little bit of endosperm, you call them albuminous seeds, or you also call them endospermic seeds. Okay, you call it albuminous seeds or you call it endospermic seeds. Fine, agree? Okay, now examples, that is more important. Examples for non-albuminous, you have pea, you have gram, you have groundnut, all of them where there are two cotyledons. If you try to break the groundnut, you can find two pieces, right? So basically pea, gram, groundnut, all of them, they are dicots. Talking about albuminous seeds, the ones that have they are monocots. Example, what do we have, children? Wheat, maize, rice, okay, and then castor also, if I remember. They are all coming in this category. Agree? Now, there is another type of plant, there is another type of seeds as well, where what happens, basically this is the ovule, right, and the ovule will be having nucellus, some cells which are known as nucellus. Before the formation of embryo sac, before anything is formed, there are certain cells which you call as what? Nucellus, mass of cells, and one cell among them is the one that is going to undergo meiosis and then form a megaspore, and this megaspore is the one that forms the embryo sac, which will be having the female gametes and everything. So this nucellus usually goes off, but sometimes it may persist there itself. Okay, it may persist there itself, and the one where this is persisting, what do you call this type of seeds as? You call this type of seeds as perispermic seeds, and you call this particular layer which is outside, okay, this layer which is there, the nucellus layer which is there, you call this as perisperm. Perisperm. Now, what is the example of this? Beet. The example of this is beet and black pepper. Okay, now if you are not able to understand this, I can draw and show you. So basically what happens, children, if this is the ovary, inside the ovary you have the ovule, and ovule initially has so many cells, bunch of cells like this, which you call as nucellus. Now for example, one among them will now be undergoing meiosis. So first meiosis 2 cells, another meiosis 1, 2, 1, 2. Okay, so meiosis has 2 rounds. First meiosis you get 2 cells, another round you get 4 cells. Among them, 3 of them will get degenerated, 1 will survive, and this one will be forming the embryo sac. Embryo sac as in this particular cell that you see over here. I am making it big. Now it's inside nucleus, okay, sorry, it's inside the ovule. So instead of nucellus cells that is over here, what do we have, children? We have embryo sac which is going to form now. That one cell with nucleus, it is going to divide itself into multiple nucleus. Almost 8 nuclei will be there, and this 8 nuclei will arrange themselves into 3 cells over here, 3 cells over there, and 2 cells over here. In between this, okay, I'll just clean it up. Okay, 3 cells over here, 2 nuclei over here. So total you have 8 nucleus condition. So it is formed because of the nucellus, 1 cell in the nucellus. Now I am saying in some ovule the nucellus still persists, and this nucellus is going to give nourishment also, and you call this layer as perisperm, which is there. Okay, so basically in the seed you will be having perisperm. Okay, that layer is perisperm, and the example is beet and black pepper. Now this is what is exactly written over here. Let's try to read it once. So in angiosperms, the seed is the final product of sexual reproduction. You know that because ovule is going to form the seed has the embryo. It is often described as a fertilized ovule. Can I say that seed is a fertilized ovule? Yes, because fertilization has happened, which is why it has now become a seed. Now, seeds are formed inside the fruits, agree, because ovary is the fruit, so seed is formed inside the fruit. A seed typically consists of seed coat, cotyledons and an embryo axis. We will come to that. The cotyledons of the embryo are simple structure, generally thick and swollen due to storage of food reserves, as I said. Okay. Mature seeds may be non-albuminous or ex-albuminous, as in they do not have albumin. As in they do not have the endosperm. Okay. Non-albuminous seeds have no residual endosperm, as it is completely consumed during embryo development for the formation of what children? For the formation of cotyledons, 2 cotyledons. Example is pea and groundnut, both of them they are dicots. Now, albuminous seeds retain a part of endosperm as it is not completely used up during embryo development because they are just making 1 cotyledon over here, and they are known as monocots. Example is wheat, maize, barley and castor. Occasionally in some seeds such as black pepper and beet, remnants of the nucellus are also persistent. This persistent nucellus is known as perisperm. This is what I exactly said. And the structure of the ovule we studied in the previous session, right? We studied in the previous session both dicots and the monocots. They have different type of structures. So if you want to have a gist of that, I can explain it over here. Okay, so let's read about this first, and then I will explain you about the structure of a normal seed. So integuments of the ovules harden as tough protective seed coats. The micropyle remains as a small pore in the seed coat. This facilitates entry of oxygen and water into the seed during germination. So the micropyle which was there, it's still there. So initially I will tell you how does the seed look. So basically the ovule was this, and the ovule had 1 inner integument and 1 outer integument, and this area was the micropyle. So they are saying now that this integuments have now hardened, and now it is going to form a seed coat. So now it will be almost looking like this. So children, if this is the ovary, okay, you will be having 1 inner layer, you call it as tegmen, okay, and then there will be 1 outer layer as well, which you call as testa. Is it visible to you? Is it visible? Yeah, it is testa. Now this area will actually be an open area. This area that you see, it's an open area, and this is still a micropyle, and this micropyle allows the entry of things and exit of things. Okay, fine. Now both of them together, I say that it is going to form the seed coat. They are going to form the seed coat. Going ahead, as the seed matures, its water content is reduced and seed becomes relatively dry, 10 to 15% moisture by mass. The general metabolic activity of embryo slows down. The embryo may enter a state of inactivity known as dormancy. So they are saying that seeds as they mature, what happens? Basically they could enter something which is known as dormancy, where the metabolic activities, as in whatever activities for the formation of things, for the breakdown of things, whatever is there, all the activities basically they slow down. Slows down. Okay. Why it could happen? It can happen generally because of genetic reasons or due to environmental stress. For example, there is no enough water. There is no enough water for the plant to grow. At this point of time, if the seed decides to germinate, obviously it won't be able to survive. So there will be other hormones coming in action such that Now the seed will be under dormancy. See, because of stress hormone being active, for example abscisic acid and all of them getting active, this particular seeds to remain dormant. And in this dormant state, children, 10 to 15% of what? 10 to 15% they become relatively dry. That is what? 10 to 15% moisture biomass. That is the thing. So 10 to 15% moisture biomass is reduced basically in them when they are entering into dormancy. Now going ahead, as ovules mature into seeds, the ovary develops into fruit. We studied about that. The transformation of ovules into seed and the ovary into the fruit proceeds simultaneously. The wall of the ovary develops into wall of the fruit known as pericarp. So this was about your ovule. This is what, children? Your ovule, which is basically now known as the fertilized ovule. Okay, so this is basically, I just messed up. This is about the seed which is known as the fertilized ovule. Now outside the seed, what do we have? We have you have this structure over here. What do you call? I don't want to, okay, this structure over here, which is the wall of the ovary, which is the wall of the ovary. Your ovary is there, which is going to form the ovary, is going to be the future fruit. Agree? And its wall, you call this as pericarp. Now do not be confused between perisperm and pericarp. Pericarp is the outer layer of the ovary. Okay, outer layer, outermost, basically the wall of the ovary, you call it as pericarp, and pericarp again will have division. Pericarp again can be divided into few layers. Okay, so there will be, I will show you. So basically, if I say that there is mango. Okay, in mango there is an outer layer. This outer layer you call it as epicarp. Okay, and then there is a very important layer over here which we eat. And what do you call this layer which we eat, the pulpy layer that you eat? It is known as mesocarp. Okay, and then there is a innermost layer which you call it as a seed, but seed is inside this innermost layer, and this innermost layer is known as endocarp. So this is all about the walls of the ovary. Together 1, 2 and 3. Together 1, 2, 3. What do you call it? You call this as pericarp. Now if you are wondering, what's, ma'am, where is the seed now of the mango? So basically mango seed will be over here. Okay, the seed will also have its own covering. As I said, there will be testa, and there will be what, children? Tegmen over here. So that is something inside this particular thing that is known as endocarp. So seed is entirely like that. I am talking about the wall of the ovary, and that is known as pericarp. Now sometimes it looks like this. You call these fruits as fleshy fruits. What do you call them? You call them as fleshy fruits. Now there are times where the pericarp is not advanced like this. It could be dry, it could be papery. In that case, you call that particular fruit as what? Dry fruits. You call them as dry fruits because they do not have the pulpy content over there. Every outer layer is what? Very papery. It is very thin layer. Okay, so that is something different. This is something different. Fine. Okay, so we in this particular slide studied that the ovule, okay, the fertilized ovule, the seed, what you call, has two layers. The integuments have gone. Now you have two layers. One is tegmen, which is the inner layer, and one is testa, which is the outer layer. Okay, so test the vigor, so that is why you have testa outside. You can think like that, to test the strength of this particular ovule wall. Okay, now one end of it will be open. It is known as micropyle. Okay, and this micropyle will be needed for exchange of things. Now going ahead, there is ovary around it, and this ovary also has a wall. You call them pericarp. Okay, you call it pericarp. Pericarp can be divided into three: epicarp, mesocarp and endocarp. Epicarp is the outermost layer, okay, which is thin. Mesocarp is the fleshy layer and endocarp is kind of thick layer which is there inside, and inside that you have the ovary and all the stuff. Agreed? Now, yes. Okay, so the fruits may be fleshy as in guava, orange, mango, etc., or may be dry as in groundnut, mustard, etc. So groundnut and the mustard, basically they have a dry thing outside, and this will be the pericarp in total. So it will be very dry, papery, whereas it will be fleshy in the case of guava, orange and mango. Agreed? Now, do we have anything? Many fruits have evolved mechanism for dispersal of seeds. Recall the classification of fruits. Okay, one question. Is there any relationship between the number of ovules in an ovary and the number of seeds? Now children, the ovules itself are going to convert into seeds if fertilization has happened, right? The ovule itself will be getting converted into seed if fertilization has happened. In usual cases I am talking, okay. So in that case, if the number of ovules are 2, the number of seeds can be 2 or less than 2, because I don't think that all the ovules can undergo fertilization. There are chances that it might not have. So it can be 1 also, it can be 1 or it can be 0, okay, or it can be 0. So if seeds are there, then obviously it should be either same number of the ovule and the seeds, okay, or it can be less than the number of ovule, because all ovule might not undergo fertilization to form the seed. Okay, I hope this is also clear to you. Now, what is important? One more thing I must say. Yes, talking about coconut. So, in the case of coconut, if you see, if you have eaten coconut, you know, this green color thing that you see, this will be the epicarp, children. Now, inside this green thing, you have, if you open, you will get the coconut thing. But there is husk of coconut as well over here, okay, the white color thing which you do not eat. Now, over here it's not fleshy, it's not something that you eat. So, this is mesocarp. I am just giving an example that always it's not like that you can eat the mesocarp. There are chances where you cannot eat the mesocarp. Now, this innermost layer that you see over here, this is the epicarp, which is very hard structure, okay. And now inside the epicarp, then you have the seed, which has the covering and everything. It has the covering, it has the embryo, all the things. Embryo, sometimes it's there, sometimes it's not there. Okay, fertilization happens, it's not there. So basically, it takes time for the formation. But yes, it has the seed inside the coconut. Inside the coconut, it has the seed. All that you see is known as the pericarp. Okay, so that's there. And now what are we left with? Okay, now seed, when you talk about seed, as I said, so seed will be containing seed coat. That is this layer that you see over here, the testa and the tegmen. Then, as I said, it might contain the this thing, cotyledons and the embryonal axis. Yesterday also I taught you. So there is embryonal axis. I'm going to show you over here only. So I'm making a dicot seed. So it has the 2 cotyledons, and this is the embryonal axis that you see over here. The lower part or below the cotyledons will be forming the epicotyl. Yesterday we studied, so I am not making again. And the above part is known as, the lower part is known as hypocotyl, hypo as in below, okay, and the upper part will be known as epicotyl, and the epicotyl will be basically having at the tip the plumule, which will be forming the shoot, and then over here we have the radicle, which will be forming the root, okay, and these cotyledons will be providing all the nourishment for this growth of the plumule and the radicle later on, okay. Sorry, this cotyledon, if I said radicle before, I meant cotyledon. This cotyledons that you see over here, it will help in the growth of this embryonal axis. Basically, it will help in the growth of the plumule as well as the radicle. That's all. So that's all over here as well. Going ahead, persistent nucellus in the seed is known as tegmen, chalaza, perisperm, hilum. You have to find your own answer. It's answer number 1. Question number 1. Give your own answer in the comment section, and then later at the end of the session, definitely I will also be solving, but I want you to try once. Okay. Now I'm missing out something, children. Yeah. So basically, you might have seen in the seeds, in the case of seed, they also have one dotted structure. You call this dotted structure as what, children? Near the micropyle, you have something that is known as hilum, the scar of the funicle. The scar of the funicle. Okay. Going ahead, what do we have? Just a second. It's too. Okay. Too cold for me. Yeah. Viability of the seeds. What do you mean by viability of the seed? Basically, its ability to germinate. Viability of seed basically means its ability to germinate. Germinate as in the plumule and the radicle, it forms beyond, and then you have a plantling, small seedling over there. Okay, that is what you mean by viability. Now, in few species, the seeds lose the viability within a few months. Okay, that can happen. Seeds of a large number of species live for several years. Some seeds can remain alive for hundreds of years. There are several records of very old yet viable seeds, and that is important. You have an example of something that is known as lupine. Okay, the scientific name is Lupinus arcticus. Lupinus arcticus. It was found in the Arctic tundra. Okay, it was found in the Arctic tundra, and the seed germinated and flowered after an estimated record of 10,000 years. Germinated after what, children? Germinated after 10,000 years. Is this a small number, children? No, it is not at all a small number. Now there is one more thing that is given over here. So there is a recent record of 2,000 years old viable seed, which is date palm. Okay, and the scientific name of this is Phoenix dactylifera. Okay, the scientific name is Phoenix dactylifera. For how long it has remained? 2,000 years. So over here 10,000, over here 2,000 years. These are very much important examples that you need to remember. Okay. Now it was discovered during the excavation of King Herod's, as in when Jesus Christ was there at that point of time. Okay, Herod's palace near the Dead Sea. Okay, I hope you got it. So it's not like Jesus Christ was there at that time. No, that's not what I was saying. I was saying that it was done at the excavation at the King Herod's palace. So King Herod was a place and is a biblical character. Okay, who was there. Now that's all. That's all the viability of the seed. Now there is a mechanism to check the viability of the seed, which is the next question that I am having over here. I'll try to explain you this question because it's not there in your NCERT. The viability of seed is tested by this, and the answer is this: 2,3-triphenyl tetrazolium chloride. So what happens when you put 2,3-trichlorotetnafluorenzolium on the seeds? Okay, basically it will turn pink. Okay, why does it turn pink? Because it shows that certain metabolic activities are happening in the seeds, and this basically the seeds are going to release some hormones or probably some enzymes, which is why it would turn pink in color. So this is the correct answer, children. Please remember it. You write it somewhere. It's important. Came in NEET 2013, completely out of box question. Okay, going ahead. All those seeds. Okay, we are going to talk about Apomixis and Polyembryony. Let's start. All those seeds in general are products of fertilization. So we as of now have studied that only fertilization happens, then only we can get a seed. Agree? I told you that. Now what happens? A few flowering plants such as some species of Asteraceae and grasses have evolved. Just a second. Asteraceae and grasses have evolved special mechanism to produce seed without fertilization. Now they are saying we can produce seeds without fertilization. We do not need help. Your help, and this process is known as apomixis. Please remember these examples, small examples. So Asteraceae and grasses, children, both of them, they are definitely going to show what? Apomixis. Okay, now what happens in apomixis. Thus, apomixis is a form of asexual reproduction. Can I say it's an asexual reproduction? Because over here, I am not taking the help of fertilization. And if I am not taking the help of fertilization, there is no fusion of gametes being formed, then obviously it is asexual reproduction that mimics what? Sexual reproduction. So, this is the process that is actually asexual reproduction, but it is mimicking what? Sexual reproduction. Why do I say it's mimicking the sexual reproduction? But over here, because we are producing seeds. Now, seed is the end result of sexual reproduction, right? But actually what is happening? Asexual reproduction is happening. There are several ways of development of apomictic seeds. Let's see. In some species, the diploid egg cell is formed without the reduction division and develops into the embryo without fertilization. So, basically what happens? Usually first condition, this is the ovule and the female gamete is there, the synergids are there, the antipodal cells are there and the free nuclei is there. Over here, this is your female gamete and all of them usually are haploid, as in there are n number of chromosomes, half the number of chromosomes, so that when the male gamete, which is also carrying half the number of chromosomes, when both of them come together, then n plus n, you have 2n number of chromosomes and everything is sorted. But they have decided that they do not want to undergo fertilization. So, if they do not want to undergo fertilization and if you want 2n, because obviously you need the number of chromosomes just like any other cell, right? So, it has to be diploid anyways. So, what happened? Probably what might have happened is that this egg cell is formed without meiosis. Usually this happens because meiosis takes place. Because meiosis takes place, that is a reductional division, which is why the diploid 2n number of cells will now form what? Haploid cells, and that's how it is all formed. But over here I am saying that meiosis might not have taken place. In that case, all the cells will be 2n, 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 2n and 2n. So, meiosis has not taken place. All of them children will be what? Instead of being haploid, so this all things are not going to happen. Instead of being haploid, now I am going to write in white over here, it is going to be diploid. It is going to be diploid. And now this egg cell which is there will undergo division by itself and form the embryo and everything. When embryo is formed, obviously it is forming a seed. If embryo is formed, obviously this whole structure will be now known as what? A seed. So, this is a very unique thing. It does not happen usually, but this is happening. Okay. This is happening because of some hormones, because of something that is there in the case of plants. Okay. Now, second case, now this is one case where the diploid egg cell is formed without reductional division and develops into embryo without fertilization. More often, as in many citrus and mango varieties, some of the nuclear cells surrounding the embryo sac, starts dividing, protrude into the embryo sac and develop into embryo. So, basically this is one condition where the female gamete is getting diploid and now this particular is going to form the embryo. The second condition that we have over here, I am going to make over here. Okay. Second condition is when, again I am making, so this time there is the ovule, and ovule, as you know, it had nucleus cell. Okay, and this nucleus cell later it has the embryo sac being formed. So, I am saying the nucleus cell is still persisting over here. Okay, and the embryo sac is being formed over here. Okay, where you have the female gametes, you have the female gamete, you have the synergid, you have the antipodal cell and everything. But now I am saying this nucleus cell which is there, it is going to divide itself and get inside the embryo sac. It is going to get inside the embryo sac, and in this way each of them that you see over here, each of them are going to form the embryo. Each of them are getting inside the embryo sac. Okay. So, I am writing cells of nucellus gets inside the embryo sac and now will be forming embryo. But these cells could get in, then these cells could also get in over here. This can also get in. So in this case, basically 1, 2, 3, 4, 5, how many embryos are there? There are so many embryos. So you call this condition as polyembryony. You call this condition as polyembryony. And where can you see this, children? What are the examples that are given in your book? Your examples are citrus fruits and also mango. And also mango. So now it would be giving rise to multiple embryos. Okay, this is known as polyembryony, and this happens because of the second type of apomixis, where the nuclear cells are going to get inside and form the embryo. Why do I still call it apomixis? Because over here is fertilization happening, children? No. The seed is being formed without fertilization. The seed is still being formed without fertilization. Why do I call it a seed? Because embryo is being formed inside, right? And over here in the second type of apomixis, polyembryony is seen. Multiple embryos are seen, and this is seen in citrus and mango. This type of apomixis is done by citrus and mango plants. Okay? Done? This is what is exactly written over here. So first case is where the diploid egg cell is formed without reduction division. That is this thing. Second is where basically nuclear cells surrounding the embryo sac starts dividing and protrude into the embryo sac and develop into embryos. In such species, each ovule contains many embryos. So many embryos are there. Occurrence of more than one embryo in a seed is referred to as, children, polyembryony. Take out some seeds of orange and squeeze them. Observe the embryos of different sizes. Okay, so they are asking you, observe the embryos, okay, embryos of different sizes and shapes from each seed. From where? If you take the seeds of the embryo and if you squeeze the seed, then you will be finding so many different type of embryos which will be there inside the seed. Now, if you put the seed, you will be getting different plants as well. Okay. Okay, because orange is a citrus plant, which is why it is showing that. So very important example. Please remember the names of the examples. Okay, so over here Asteraceae grassus is there as the species. Over here citrus and mango is there, which is showing the second type of apomixis with the help of the cells of the nucellus, which is also showing polyembryony. Yeah, works? Understandable? Going ahead. Okay, adventative embryony in citrus is due to adventative embryony. What do you mean by adventive, not adventative? It's my fault. Adventive. This condition is known as adventive embryony, where multiple embryos are being formed over here inside the ovule. Okay, inside the embryo sac. Now, it is due to what reason? Okay, when basically embryo, it's adventive means when basically embryo is formed without the natural process of happening it, okay, without the sexual reproductive process, it's occurring in somewhere else, without the help of without the help of your embryo sac, your female gamete, something is being formed, embryo is being formed from some other cells. You call this as adventive embryony. Okay, so here nuclear cells are helping over here to form the embryo. It's not the female gamete or the male gamete which is coming and fusing and form the embryo. I hope you got it. When we say adventitious buds, so that is the buds which are not normally formed. So adventitious buds you might have studied over here. Normally in the notes you get buds, and these buds are the ones that grows into stem or leaf or whatever. But in the case of bryophyllum, that is a leaf, the buds arises from the notches and this buds falls down to form a new plant. So that is what? That is asexual reproduction that is happening, and these buds are not supposed to be there. It's adventitious buds. Okay, something unnatural is happening over here. So whenever the adventive or adventitious word comes, just understand this is the thing that we are trying to say. Okay, now don't you think that all the embryos that will be formed, they will be all the same type, just like the parents? Will there be any genetic variation, children? If you see over here or here, will there be any genetic variation? I do not see fusion of gametes, and if fusion of gametes is not there, I cannot expect genetic variations to be there, because if both male and female characters are coming, then only variations can come in. Or basically during the when DNA replication happens, during some errors, it can occur, but that is very limited. So over here I won't be able to see much variations. I can call these things embryos which will be formed, I can call them as clones of their parents, which will be the genetic makeup is entirely what? Almost same. Okay, Now going ahead, this is the question that I have. This is question number, okay, so this was question number 2 and this is question number 3. Okay, going ahead, what is common between vegetative propagation and apomixis? This is 4. You can try to find the answer and tell me in the comment section or the chat box as of now. Okay. Next thing, nucellar polyembryony is reported in species of dash. Okay. Going ahead, types of fruits. Now that's the last topic for this. Okay. No, that's not the last topic. We have artificial hybridization as well. Yes, my fault. Let me see if I space. Yeah. In most plants, by the time the fruit develops from the ovary, the other floral part regenerates and falls off. However, in a few species such as apple, strawberry and cashew, the thalamus also contributes to the fruit formation. Now, this is a very interesting topic for me. So, usually, as I said, usually what is the case? We have the stigma, we have the style, and this is the ovary. Okay. This is the ovary, and this ovary is the one that will be forming the fruit. This type of fruits are known as true fruits. I call them true fruits. Okay, this is condition number 1. Second condition. Second condition is where, you know, we have something like this. This is thalamus and this is pedicel. Okay. And children, this will be the flower parts, the petals and everything, and you have the whole thing over here, that is okay. So basically in this type of plants, okay, in this type of plants, what will happen if I can make anther also? Let's make anther and the stamen, anther and the filament as well. Now, in this type of plants, along with the ovary, the thalamus also combines together to form the fruit. So, who all are combining together? Over here, I see thalamus plus ovary contributing to the formation of a fruit. Now, these type of fruits, I call them false fruits. What are the examples, children? Apple. The apple that you eat is the commonest example of this thing. Example, apple. And then cashew. And then what? One more thing is there. Strawberry. All of them, they are example of this thing that is false fruits. Okay, where the thalamus is also contributing for the formation of, which is why we won't be able to see anything. It's like, and rest of things, they obviously degenerate. Okay. Now, there is a third variety. That is the epic variety, which is known as Parthenocarpic fruits. So, we have studied where the seeds are formed, where the embryo is formed without fertilization. Okay. Where the embryo is formed without fertilization. Now, this is where fruit is going to be formed without fertilization. Fruits are formed without fertilization. So, apomixis is where seeds are formed without fertilization, fruits are formed without fertilization in the case of parthenocarpic fruit. So, over here what happens? Basically, for suppose there is an ovule, okay, and there is the ovary. The ovary which is there, it will now make a fruit even if no fertilization took place. Now, usually what happens, I'll tell you. Usually the ovule is the one that releases certain hormones, okay, it releases certain hormones which helps in the formation of fruit over here in the ovary. But over here, okay, due to the activation of certain stress hormones or due to some genetic variations that is happening over here, the ovary itself is going to produce certain hormones like auxin, gibberellin, and everything, such that the ovary will now be converted to a fruit. Such that the ovary which you see over here will now be converted to a fruit. So, this is happening because of some hormonal changes which is happening inside the whole plant body, okay, over here. This is known as parthenocarpic fruit. So, do you see if fertilization has not taken place, do you think that there will be seed? Do you think there will be seed? No. So, in parthenocarpic fruits, no seeds are there. No seeds are there. So, it can happen naturally, children. It can generally happen naturally, but why to make it happen naturally? When you can inject auxin. You can inject auxin and make parthenocarpic fruit. You inject auxin such that the ovary will now be converted to what? Directly a fruit. And this happens in the case of, where should I write it? In the case of banana. So, banana basically are seedless because of the same reason. It's not actually seedless, but you can see small seeds that is usually there, but sometimes, mostly when it is made like this, you don't find seeds over there. So, they are seedless, okay. Apart from banana, you have seedless tomato and things like that. So, whenever things are seedless, just imagine that they are what, say, parthenocarpic fruits, okay. Example is banana. I am telling you again, over here, the ovary is not forming a fruit. Apomixis is where the ovule was forming a seed without fertilization. Here, it is where ovary is forming a fruit without fertilization. Okay. I hope this is clear. Now, let's read it. It will be more easy for you to understand it. Okay. In most plants, by the time the fruit develops from the ovary, other floral part degenerates or falls off. However, in a few species such as apple, strawberry and cashew, etc., the thalamus also contributes to fruit formation. Such fruits are known as, children, false fruits. Most fruits, however, develop only from the ovary are known as true fruits. Although in most the species, fruits are the results of fertilization, there are few species in which fruits develop without fertilization. As I said, such fruits are known as parthenocarpic fruits. Banana is one such example. Parthenocarpy can be induced through the application of growth hormones such as auxin and gibberellin, such that such fruits are what, children? Seedless. They would not be having seed as fertilization has not taken place. So, these are the three different type of fruits that you need to remember along with their examples. Okay, works? Going ahead, what do we have? Artificial hybridization. Now, this is a topic, extended topic, which was there in your NCERT. I was thinking of leaving it, then I thought that I should be, it's just a topic, why not explain it? So, a breeder is interested in crossing different species and often genera to combine desirable characters. I want desirable characters to be there, okay. So, I want superior varieties, something that has more desired quantities that I wish to have, okay. So, artificial hybridization is one major approach of crop improvement or program. Artificially, you are hybridizing. Hybridizing, it's a mix of two things, okay. Now, over here, in such crossing experiment, it is important to make sure that only the desired pollen grains are used for pollination. Obviously, if some other, for example, okay, again I have to make this, but it's okay. So, we have the thalamus, we have the pedicel, and then we have the flower, which has the ovary and also the anther, okay. So, now if I want to produce a hybrid variety, obviously I cannot make the pollen over here to land over here. In that case, it will be the same thing happening, self-pollination would be happening. I want a hybrid variety to take place. Over here, what could I do? So, for that, the first process is known as emasculation. So, the first process that you see over here that we are going to do is what? Just a second, children. Emasculation. What happens basically in emasculation? Basically, if the female, where is it? Okay, if the female parent bears bisexual flowers like this, as in when they have two sexes together, removal of anthers from the flower buds before the anther dehisces using a pair of forceps is necessary. This step is known as emasculation, where basically you are going to remove the anther such that it won't be producing, if it produces pollen grains, then obviously it would go land. I do not want this to happen. So, what do? I remove this instead, okay. Basically what? I remove the anther in general. Done? Let me remove this also. Over here, going ahead, this is known as emasculation. Now, again what will happen, children? there will be pollen grains all around, okay. So through wind, pollen grains can come over here and land over here. I do not want that also. I do not want this to happen. So to protect it from anything, what do? I cover this whole thing. And this process is a second process which is known as bagging. Now, emasculated flowers have to be covered with a bag of suitable size, generally made up of butter paper, to prevent contamination of its stigma with unwanted pollen, something that we do not want. Now, this is known as bagging. When the stigma of the bagged flower attains receptivity, now it's mature enough, okay. It is receptive. Mature pollen grains collected from anthers of the male parent are dusted on the stigma. So, we have different type of roses. And we have two similar type of roses, but one is giving good type of flowers, the other one is not. Now, I want the one that is giving me good type of flowers over here. So, I collect pollen grains from them, and then what do? I basically put this pollen grains which are of high variety over here. This is king pollen which is I am putting over here, okay. So, that is the next step. So, that's all. So, we have 1 and 2, and the next step is transfer of desired pollen. And then tada, the whole fertilization and everything will happen, and now it will be giving rise to seeds. And the seeds, if I put somewhere, basically I can give rise to new plants with what? Good variety. So, hybrid varieties of plants can be formed with the help of this, and this is known as artificial hybridization. It's not occurring naturally, it is artificially happening. So, that's all, children. That's all for today. I couldn't find question for the same, okay. For artificial hybridization, rest for everything we have a question. Let's try to see who all got your answers correct. First thing, persistent nucellus in the seed is known as. So, nucellus is there, it is persistent, and it is going to provide nourishment if needed. This will be known as, children, perisperm. Example is very much important, children. Black pepper, and then what do we have? Beet. These are the example. The viability of seed is tested by 2,3,5-triphenyl tetrazolium chloride. I already told you this answer. Adventive embryony in citrus is due to. Why do we have embryonic condition with not natural embryos that are being formed over here? What is the reason for it? The nucellus. Because the cells of the nucellus are going to enter the ovule, and then embryo sac, basically, not the ovule, nucellus is there in the ovule. So, the cells of the nucellus are going to enter the embryo sac, and there will form multiple embryos. Polyembryonic condition will be there. This is citrus plant, okay. Citrus plant, they will be showing this thing. Now, next thing, if it was written mango, still you had to understand that it was nucellus. What is common between vegetative reproduction and apomixis? Both are applicable to only dicot plants, is like that? No. Both bypass the flowering phase? No. Both occur around the year? No. Both produce progeny identical to the parent? Yes. Vegetative propagation, where you are using the vegetative parts, you are not doing sexual reproduction there. It is also asexual reproduction. As I told you, the bryophyllum leaf, it is forming buds over here, and these buds are going to fall off. Okay, there is no fertilization, nothing is happening, and these buds, when they fall off, they are going to form a new plant over here. Or cutting or grafting, which we see in roses and sugarcane and stuff like that. All of them, it's known as vegetative reproduction, where you are not allowing them to fuse or do anything like that. No sexual reproduction is happening there. It's asexual reproduction. So, apomixis and vegetative reproduction, basically what do you have in common? The progeny, both of them obviously will be identical to the parent, as fertilization process is absent. Okay, apomixis is what? Production of seed without fertilization. Okay, now nucellar polyembryony is reported in species of, same thing, because of the nucellus, polyembryony is happening. This will be observed in citrus plants, student, we studied right now. Yeah, so that's all. Tell me who all got it correct. I'll be so happy seeing your comments down there. Yeah, so that's all, children. Apart from that, there's one question for my dear little champions. Let's see if you would be able to do it. Perisperm differs from the endosperm in being a diploid tissue, its formation by fusion of secondary nucleus with several sperms, being a haploid tissue, having no reserve food. So, this question answer we will be discussing tomorrow, but if you have already got the answer, then you can put it down in the comment section. Yes, Works? And do let me know who all got the answers correct. Now that's all, children. If you think that this particular video was helpful for you, then do not forget to subscribe to the channel. And also one more very important thing, we have the weekly mock test day after tomorrow, so please study for that. On Sunday we have it. Okay, I'll include all the questions of sexual reproduction in flowering plants, and if you want me to put questions from other chapters as well, do let me know in the comment section. I'll be definitely putting them. So whatever chapters we studied previously, if you want a few questions from those chapters as well, do let me know. I'll be putting down in the question paper, but you have to let me know for that. Okay, so put down in the comment section. That's all, children. Do not forget to subscribe to the channel and have a really good day. Do not forget to smile. Bye-bye.